Welcome to Flow Cytometry Fundamentals, how cytometry can be used to answer your research questions. I'm Vanta Jameson, Manager of the Peter Doherty Flow Cytometry Facility here at the University of Melbourne's Cytometry platform. The aim of this video is to introduce researchers and students who may have limited background in flow cytometry to this critical technology. I will take you through what flow cytometry and cytometers are, the types of particles that you can analyze, and the information you can retrieve and applications that are commonly used. You will learn what fluorescence is and critical controls that must be supplied. Then I'll take you through how cytometers work, how to interpret cytometry data, and end with how a major application, immunophenotyping, works. Flow cytometry, or simply cytometry, is a technique used widely in clinical laboratories to diagnose and track blood cancers, in the pharmaceutical industry for drug development and screening, in life science labs within universities and medical research institutes, and even on ocean-going research vessels. Researchers in fields as diverse as immunology, stem cells, neural and reproductive biology, cancer, hematology, plant, marine and microbiology, nanoparticles and material sciences all make use of cytometers. In biology, cytometry is used to analyze mostly single cells ranging from half to 30 micrometers in size in heterogeneous mixes. Cytometry provides information about their phenotypes and frequencies by measuring physical properties and fluorescence. Particles typically analyzed at the Melbourne Cytometry platform come from mammalian sources, including cell lines and primary samples from humans and rodents. We analyze erythrocytes and leukocytes from blood and hemopoietic organs such as the spleen and lymph nodes, and various cell types from dissociated solid organs such as skin, liver, lung, gut, muscle, the eye, and tumours. In addition, we analyze cells from other vertebrates such as fish, invertebrates such as flies and worms, coral, and non-animal samples such as algae, bacteria, fungus, parasites, and pollen. As samples run through a cytometer, measurements are taken of each individual particle. When running cell samples, measurements include relative size and complexity. This information differentiates debris from cells, live cells from dead cells, the type of cell that it is, or if a particle is actually a clump of cells. It also measures emitted fluorescence, including the intensity of different fluorochromes that may have been added to the particles, and the intensity of autofluorescence or natural glow. Data collection is very fast, collecting thousands of events per second. It is quantitative and qualitative, and importantly, unbiased. Many physical and fluorescent measurements are taken simultaneously, and because there are so many events recorded, statistics are robust. Cytometers measure fluorescence, so let's define what that is. Fluorescence is the emission of radiation, in this case light, that occurs when electrons in a fluorescent molecule are excited from rest to a maximal energy level. As the electrons return to a more stable state, energy is released as heat. As electrons return to their resting stage, the final energy release is emitted as fluorescence. Fluorescence lasts for 1 to 10 nanoseconds, depending on the fluorophore, and each fluorophore can undergo many cycles of excitation and emission until they exhaust or photobleach. Each fluorophore has an excitation and emission spectrum. Shown on the right is the excitation and emission spectrum of the fluorescent molecule phycoerythrin, or PE. Its excitation spectrum is shown with a dotted line, while the filled histogram shows the wavelength of light most likely to be emitted after excitation. The coloured vertical lines are the wavelengths of laser light on a 5-laser cytometer: UV, violet, blue, yellow-green, and red. The x-axis shows the wavelengths in nanometers, and the y-axis shows the percentage chance of excitation or emission at a given wavelength. Excitation refers to the range of energy that can energize electrons to their maximal energy state. Excitation max, EX max, is the optimal wavelength of energy that causes the greatest amount of electron excitation for a given fluorochrome. For PE, we can see that UV, violet, and red lasers do not excite PE. The blue laser excites PE at approximately 65% efficiency, while the yellow-green laser excites PE at greater than 90% efficiency. Emission refers to the spectrum of light emitted by a fluorochrome as it heads back to its resting state. EM max, or emission maximum, is the wavelength of light most likely to be emitted by the fluorophore. PE is most likely to emit photons at approximately 582 nanometers, but there is a chance that longer wavelengths will also be emitted. Emitted light will always be of a longer wavelength or lower energy than the light required to excite the fluorochrome. PE is an example of a fluorescent molecule or pigment that occurs naturally in algae and cyanobacteria, allowing them to photosynthesize. Viewed under a microscope, excited by the right wavelength, these life forms will autofluoresce because of the presence of PE and other pigments and structural and metabolic components. Biological particles, including pollen and mammalian cells, have a level of natural fluorescence caused by molecules in their makeup and their metabolic activity. In some cases, autofluorescence alone can be used to identify cells of interest, but in most cases, autofluorescence is not sufficient, and extrinsically added fluorochromes are required to resolve distinct cellular populations. PE is also a fluorescent molecule that has been harnessed by developers of fluorescent reagents to bind to antibodies and probes for imaging and cytometry. This is what we call extrinsic fluorescence. There are many extrinsically added fluorochromes, a sample of which are shown here. Each have their own excitation and emission spectra that cover the entire visible light spectrum, as well as ultraviolet and infrared wavelengths. Some fluorochromes, such as PE and allophycocyanin or APC, are derived from nature, while others are synthesized small molecules and polymers, such as Alexafluor 488 and Brilliant Ultraviolet 395, respectively. Fluorochrome options are also extended by the binding of two fluorochromes to produce tandems. In tandem fluorochromes, such as Brilliant Violet 711, a donor fluorochrome is excited at a higher energy wavelength than its partner or acceptor. The donor's emission excites the acceptor fluorochrome, and the acceptor's emission is the one that is measured. In cytometry experiments, many fluorescent molecules are combined into a single tube of cells by bonding them to antibodies that bind molecules on cells, but more on that later. To date, successful 50-color fluorochrome panels have been published. On top of this, heterogeneous cellular mixes can have cells with different levels of autofluorescence, meaning that each experiment can be very complex with a lot of excitation and emission of fluorochromes occurring simultaneously. Now back to cytometers. Flow cytometry is performed on cytometers, including analyzers and cell sorters. Analyzers, on the left, identify and quantify particles in samples, and generally, assays are performed by researchers after hands-on training has been provided. Sorters, on the other hand, identify and quantify particles in samples, but also allow the retrieval of up to 6 pure populations simultaneously for onward workflows. Retrieved cells are frequently put back into culture for functional assays or used for adaptive transfer and transplant, cloning, imaging, and the extraction of genomic material or proteins for various omics. All cytometers are made up of 3 major components: the fluidics, optics, and the electronics and data display. The fluidic system, which is made up of sheath tanks, tubing, regulators, pumps, filters, valves, and the flow cell, regulate the flow of samples within sheath fluid, ensuring a constant and predictable passage of particles through the cytometer. Optics include the lasers, fiber optic cables, mirrors, optical filters, and detectors. Lasers provide the light source that is scattered and excites fluorescent molecules. Scattered and emitted photons are split into discrete packets of photons of similar wavelengths by mirrors and filters before they are amplified at detectors. The photons produce an electrocurrent within the detectors that is proportional to the initial input of photons that make it to that detector. Finally, the electronics process and allocate signals amplified at each detector to discrete particles. The signals are digitized and presented as events on bivariate plots and histograms representing each measurement. Here is a schematic of how a cytometer works. Your sample is loaded into the sample input point where it is carried by sheath fluid, either water or saline, into the flow cell and past lasers. The action of particles passing lasers causes light to scatter or fluorescence to be emitted. This scatter and emission are captured and measured by detectors, and the data is presented in the form of histograms and bivariate plots. Analyzed particles are carried to the waste container. If using a cytometer with cell sorting function, populations of interest can be selected from the plots, and particles within these regions are deflected into collection vessels while non-desired particles are taken to waste. Data is represented in the form of histograms and bivariate plots. Histograms, shown in the top right corner, provide information about the number of events of one physical or fluorescent measurement of the particles. Bivariate plots, in the bottom row, provide information about two measurements of the particles on the x and y axes. For all measurements, the further along the axis a point is, going to the right or heading up, the more fluorescent, larger in size or higher in complexity a particle is. On the dot plot on the left, we're looking at two physical measurements of leukocytes, relative size represented on the x-axis and complexity on the y-axis. Each dot represents a single particle that has been measured. Particles with similar measurements are clustered together. We can see debris in the bottom left corner and distinct clusters of particles representing lymphocytes, monocytes, and granulocytes that go from lowest to highest size and complexity respectively. The plots on the right are examples of pseudo-color plots, where the color intensity of each cluster is representative of the number of events in that cluster. Clouds go from blue, the least events, to red, most events. In this plot, we are measuring the intensities of two fluorescence measurements, with the quadrants indicating the boundaries of presence or absence of fluorescence. In the bottom left quadrant, particles are negative for both fluorescence 1 and fluorescence 2. Particles in the bottom right are positive for just fluorescence 1, and particles in the upper left are positive for just fluorescence 2. Particles in the upper right are positive for both fluorescence 1 and 2. You'll notice on these plots that we are using a log scale to measure fluorescence. Measuring fluorescence on a log scale allows us to display low to high fluorescence intensity on the same scale. Comparing the particles in the plots side by side, the particles in the blue circles have the same level of fluorescence intensity for fluorescence 2, whereas the particles in the purple box on the left have roughly 10 times the amount of fluorescence intensity of the particles in the green box on the right. To get accurate measurements in cytometry, we must first ensure that we have fluidic stability throughout the entire cytometer's path. During hands-on analyzer training, we focus on priming the fluidics and assessing that the signals are stable before any acquisition experiment. Fluidic stability and laminar flow ensure that particles are optimally excited by each laser and that particles pass by each laser at a constant rate so that the electronics could combine signals produced at each laser into a single event. When sorting, fluidic stability ensures that drops break off the stream consistently and that particles arrive at the break-off point at the predicted time so that the drop carrying the correct particle is charged and deflected into a collection vessel. As alluded to earlier and simplified for this presentation, cytometers measure physical characteristics of particles, revealing their relative size and complexity. These measurements are captured based on the amount of light that is scattered as particles cross the blue or violet lasers. As cells cross a laser, light is scattered in all directions. A detector in the horizontal plane to the laser measures the relative size of particles based on the angle of light diffraction. The larger the particle, the greater the angle of diffraction. This measurement is known as forward scatter or FSC. There is also a detector at 90 degrees to the plane of the laser that measures light refraction as light bends around internal cellular structures such as the nucleus, vacuoles, and granules. The more refracted light, the more complex a particle is. This measurement is known as side scatter or SSC. Of note, though, forward scatter and side scatter measurements do not convey size or complexity when measuring very small particles such as exosomes or other nanoparticles, or particles made of different materials. The optical component of a cytometer is responsible for light splitting. The fluidics carry particles through a flow cell in single file where they cross the path of several lasers. If an autofluorescent molecule or added fluorochrome are excited by the lasers, fluorescence is transported via fiber optic cables associated with that laser to a series of detectors. Each laser has its own detector array. When light reaches the first detector in an array, bandpass filters let through light of specific wavelengths, for example, violet, blue, green, yellow, or in this case red. Light that does not match the bandpass filter's spec is reflected to the next bandpass detector pair. Photons that make it through the bandpass filter to a detector are amplified to produce a photoelectron current proportional to the original input of photons. There are two optical systems in cytometry, conventional and spectral. Conventional cytometers were developed first, nearly 50 years ago, when fluorescent probes were limited. In conventional cytometers, each fluorochrome is allocated to a laser detector pair. For example, brilliant violet 711 is allocated to the violet laser, which excites the fluorochrome at greater than 90% efficiency, and the 710/50 nanometer bandpass detector, where peak emission is measured. While BV711 emits photons that are outside of this bandpass range, both shorter and longer wavelengths, these emissions are not included in its fluorescence measurements, and mathematical algorithms are used to remove or compensate them out of adjacent detectors. In contrast, in spectral cytometers, emissions are measured after passing every laser, resulting in a spectral trace of different intensities across all laser detectors. So while BV711 is still excited most efficiently and emits most intensely after violet excitation, we can see that there are secondary, albeit lower, emission peaks in all laser detector arrays. Comparing the two traces here, the conventional trace at the top is represented on the spectral trace within the purple box. The peak emission in the 710/50 detector is equivalent to the two peaks at detectors V14 and V15. Because of the unique spectral traces or fingerprints for each fluorochrome, spectral cytometers allow for much larger fluorochrome panels to be used compared to conventional cytometers that are limited to the number of fluorochromes as there are laser fluorescent detector pairs. Regardless of whether using conventional or spectral optics, defining fluorochrome excitation and emission signatures is a critical part of setting up any experiment on a cytometer. As discussed on the previous slides, each experiment may have many autofluorescent particle types as well as extrinsically added fluorochromes within a single sample, and thus interpretation of the signals may be very difficult, if not impossible, without first defining what each fluorochrome signature is before running test samples. It is therefore critical that, as well as bringing along a fully labelled sample, that each experimental setup includes controls to define fluorescent signatures. Firstly, unlabelled particles of the type being analysed are required to set up and define baseline levels of fluorescence, that is, autofluorescence, in each detector. Secondly, single colour controls where particles are labelled with one fluorophore at a time are required to define the emission of each fluorophore by itself. For these single colour controls, it is critically important to have unlabelled particles of matching autofluorescence. For example, if using cells to define a green fluorescent protein signature, it is important to have a parental or wild-type cell of the same type that do not have the green fluorescent protein. So, how do you get fluorescence into or onto cells? Let's start with fluorescent proteins. Plasmids encoding fluorescent protein DNA are often introduced into cells to visualize and track cells in vitro and in vivo. Plasmids can be introduced into cells via transduction with a virus or via transfection, where the fluorescent protein DNA is transcribed and translated by the host cells. Flow cytometers can measure cells that express the fluorescent protein, even if it is intracellular. Dyes can be added to cells by simply pipetting the dye directly into the cell suspension buffer. Some dyes, such as nucleic acid binding dye propydiamidite, or PI, that fluoresces red, only enter cells if the membrane is compromised. PI is used to identify cells that have died in a live cell suspension. PI is also used to track cells at various stages of the cell cycle by measuring DNA content. The more DNA that a cell has, the brighter the cell is. For example, cells that are in S phase or G2M will have a brighter intensity than cells that are resting in G0 or G1. Other dyes cross the intact membranes of live cells to label DNA, mitochondria, or other proteins in the cytoplasm. CFSE is one such very stable green dye. It crosses the membrane of live cells and covalently binds proteins. When a cell divides, the CFSE is shared equally between the daughter cells and halves in intensity. Flow cytometry can track the number of divisions a population of cells has undergone by tracking the fluorescence intensity of CFSE days or weeks after its addition. Cells that have not divided will have the starting brightness of the dye, while cells that have undergone division will have less intense green signal. Immunophenotyping is the bread and butter technique of immunology and many other cell biology fields. All cells in the body have a repertoire of cell surface and intracellular markers, proteins and carbohydrates, that allow the cells to perform their specific functions. Cell surface markers, mostly referred to by their CD or cluster of differentiation or designation name, are involved in adhesion, trafficking, signaling, pathogen interactions, etc. Each cell type has its own unique repertoire of markers that can be used to distinguish it from other cell types at a much deeper level than by their physical appearance. For example, within the lymphocyte compartment, there are B cells and T cells that are indistinguishable from each other when looking at them on a forward and side scatter dot plot or by light field microscopy. However, they express very different repertoires of cell surface molecules and have very different roles in the immune system. Additionally, a deeper look at T cells reveals that there are many subtypes, including T helper, cytotoxic, regulatory, and memory cells, each again with many subtypes, based on the expression of unique repertoires of cell surface markers at various abundances and tissue distributions. In cytometry, phenotyping is achieved by the binding of fluorescently labelled antibodies specific to cell surface and intracellular markers. Antibodies bind specifically to cells that express their corresponding antigen, allowing for high-fidelity examinations of various cell types and their functions. It is often necessary for cells to be labelled with 5 or more antibodies to distinguish them from similar subtypes. Immunology and flow cytometry have grown symbiotically over the decades. Immunologists have pushed for the development of increased cytometer capabilities and improved fluorescence reagents, and consequently, our knowledge of the immune system has exploded through the ability to perform more complex analyses. There are publications and other reports that have successfully used 50 or more cell surface fluorescently bound antibodies to subtype cells in the immune system. On this slide is an example of an 8-color lymphocyte immunophenotyping experiment acquired on a cytometer. Regions have been used in sequence to narrow down to the populations of interest from a solid organ tissue. Firstly, cells were selected based on forward scatter and a live dead fluorescence marker. Live cells that have low fluorescence in a near-infrared detector were included for further analysis by surrounding them within this red live gate. These live events were next displayed on a plot showing singlets versus aggregated cells. Singlets in the blue gate were then shown on a scatter plot, and a purple gate was used to include cells of low complexity where lymphocytes are known to scatter. From there, a green CD45 positive gate marking leukocytes was selected. These leukocytes were then separated based on the expression of CD3 for T cells and CD19 for B cells. The CD3 positive events were then analysed for the presence of gamma delta T cells. Non-gamma delta T cells were then further separated using CD8 and CD4 to mark cytotoxic and T helper cells respectively. The CD8 positive T cells were then analysed for those recognising a specific tetramer. The hierarchy of progressive selection is shown on the right. B cells, gamma delta T cells, CD4 cells, and tetramer positive and negative CD8 cells were sorted for transcript profiling. Besides immunophenotyping, other common applications of cytometry include phenotyping subcellular particles, intracellular antigens, or discovering gene expression during development using fluorescent proteins as surrogate readouts, and functional assays, exploring cellular kinetics such as cell division and generational expansion, DNA content for cell cycle analysis, death and apoptosis, calcium flux, phosphorylation, protein interactions, and cytokine and growth factor production. To conclude, cytometry is a mind-blowing technology, but it is only as good as the experimental design and quality of the samples that go into it. The design phase may be extensive to select the appropriate controls and designing antibody panels and titrating reagents. As my mentor told me when I was a student, cytometry won't save a poorly designed experiment or resurrect unhealthy cells. It's garbage in, garbage out. Which leaves me to acknowledge my team at the Melbourne Cytometry platform, our platform manager, Dr. Alexis Perez-Gonzalez, and our talented and dedicated staff, Drs. Maggie Sakis and Alison Morey, Ollie Eltherington, and Tanvi Damani. I also thank the researchers with whom we've worked closely, especially the researchers at the Departments of Microbiology and Immunology and Anatomy and Physiology. Finally, I'll leave you with further reading, in particular, Introduction to Flow Cytometry, that you can link to using this QR code, and additional videos produced by the MCP team. Thank you for watching.